In our last video, we talked about the different broad categories of chemical reactions that occur within our cells. Now, we talked vaguely about energy and energy being transferred or energy being released. Now, if we're talking about energy being released, we're usually talking about it being released as heat, but it can also be released and captured and transferred. Now, in our cell, the molecule that mediates the transfer of energy in a chemical reaction is ATP. And ATP is the energy source that powers cellular work. So our cells do three different kinds of work. They do chemical work, in which they synthesize polymers from monomers. So we saw that in chapter 3, where we learned about our 4 different biological molecules and how we put them together. Cells can also do transport work, such as pumping substances across a membrane. Against a concentration gradient. In other words, active transport. So when we say transport, we are not talking about movement of molecules like a car. We are talking about movement of molecules across a membrane. So the movement that we talked about in chapter 5. Or we can or cells do mechanical work, which is the energy of movement. So we might be talking about vesicles moving along a cytoskeleton tract. We might talk about muscle movement. Or we might talk about beating cilia or moving flagella. All of these are powered by ATP, and they are powered by energy coupling. So in the last video, I talked about the idea that the energy released from an exergonic reaction can be used to drive an endergonic one. In other words, the breaking of bonds and therefore the energy release can be used to build bonds. And ATP works by releasing part of its molecular structure by breaking a bond and very often transferring that structure, forming a new bond with another molecule. So this is a diagram of our ATP molecule, and you might recognize this as a modified nucleotide. So in chapter 3, we learned that nucleotides are the monomers of our nucleic acids, DNA and RNA. And this specific molecule, ATP, is actually a modified adenine molecule. Our DNA A base. Now, a nucleotide is a 5-carbon sugar. A nitrogenous base, and a phosphate group. But you might notice that ATP actually has 3 phosphate groups, which is why we call it adenosine for adenine triphosphate, ATP. So this phosphate group, or these phosphate groups, are really important. Those phosphate groups have a lot of energy. And that energy is stored in high energy bonds. And you might have already guessed that if we break those bonds, we can release energy. So looking at this molecule, we have a whole bunch of negative charges. Now, if you think about when you bring magnets together, two negative sides or two positive sides, they repel each other. So what actually happens is this first phosphate on the end is going to get kicked off. It's pretty unstable because of those high energy bonds and because of all those negative charges. And when that phosphate group is released, we call it an inorganic phosphate, or PI, inorganic phosphate. When that phosphate is released, energy is also released because we have broken a bond. So by breaking down ATP or performing a hydrolysis reaction, we are actually going to release energy. So in chapter 3, we learned about dehydration reactions, where we build a water molecule, pull a water molecule out of the system, and form a bond. Hydrolysis is the opposite. Hydrolysis is adding water into a bond in order to break it. So we can use hydrolysis, adding water. Hydrolysis actually means to break water. So we're going to break this water into an H and an OH, and we're going to use it to split off this inorganic phosphate. Now, when we do that, we release some energy. Specific amount of energy, that is 7.3 kilocalories of energy. So every time we release ATP, we get a finite energy out of that broken bond. Now, one of the ways that ATP is used is by actually transferring this phosphate. So enzymes can use the hydrolysis of ATP to power endergonic reactions. That break up of adenosine triphosphate into adenosine diphosphate, plus a release of energy, is used to power an endergonic reaction. So in our mitochondria, powerhouse of our cell, we are actually building ATP from glucose. We're taking the energy in glucose, breaking the bonds, and transferring that energy into the bonds of ATP. But we can also break those ATP bonds. And an example of how ATP might be used is shivering. Now, when we get cold, our muscles begin to contract. And that movement requires ATP. That's why when you shiver, you shake. Your muscles are contracting really quickly. And when you break those bonds in ATP, when you hydrolyze those bonds, ATP to ADP, you also release energy in the form of heat. And that's how you warm up. So not only do your mitochondria build energy, but that energy is then used in your body for a variety of purposes, one of which is shivering. So there are three types of work that are performed in cells: chemical, mechanical, and transport. Let's talk about how ATP works in each one of these settings. The most complicated one, and the one that seems to confuse the students the most, is this idea of a phosphorylated intermediate. But don't think of it in terms of this really big word. Think about what those words mean. Phosphorylated just means that there's a phosphate involved. Intermediate means there's a couple of steps. So what this is saying is that there is a phosphate that is being transferred that is powering a chemical reaction. So ATP works by the transfer of a phosphate molecule. So a phosphate molecule is transferred from, a phosphate from ATP is transferred from one molecule to another. So in this pathway here, we are trying to build the amino acid glutamine. Now this is going to be part of a larger metabolic pathway controlled by enzymes. But in one of these steps, we're going to start with glutamic acid. Now we are building an amino acid, so that means we need to put in energy. So we are going to use some ATP, and that ATP is going to be hydrolyzed, so we're going to release a phosphate, and that phosphate is going to be transferred to the glutamic acid. So we have just performed an anabolic reaction. We've put energy in, so this is endergonic anabolic reaction. And remember that when we put energy into a system, it becomes unstable. So this phosphate makes that glutamic acid unstable. And when they're unstable, they want to react. So when this phosphorylated glutamic acid is in the presence of this amino group, this unstable glutamic acid is going to want to react. And so very quickly, it will react to form glutamine. Now in this reaction, delta G is positive, we put in some energy. That energy was this ATP back at the beginning. So just as a quick review, with a phosphorylated intermediate, one of the phosphate groups is transferred from ATP to another molecule. That molecule becomes unstable. It wants to react. It reacts with what's nearby, which if we're performing a specific metabolic pathway, we're going to have all the reactants that we need to very quickly form the product. So this phosphorylated intermediate is only there for a very, very short time before it becomes so unstable that it reacts. So ATP can also power transport and mechanical work in the cell. The top example here is actually another example of a phosphorylated intermediate, and it shows how transport proteins work. So this diagram probably looks familiar. We saw this in chapter 5, where we were moving molecules against their concentration gradient. And in chapter 5, we just said that energy is required. But the way that energy is put into the system is by ATP transferring a phosphate to that molecule, to that transport protein, making it unstable so that it causes a shape change and moves a molecule across. So a molecule comes in, ATP donates a phosphate, it becomes unstable, changes shape, and releases that molecule, therefore accumulating those solutes on one side. In transport work, ATP does the same kind of thing. It phosphorylates a movement molecule or a movement protein, so a protein that is acting as a transport molecule, and ATP releases a phosphate, binds it to our transport protein, and that causes a shape change because it's now unstable, and our motor protein, there we go, moves along. So this diagram here is a cytoskeleton tract with a vesicle. Now, when we learned about vesicles, we really just said they move through the cell, they move along cytoskeleton tracts, but we didn't talk about how they move. They move by these little transporter proteins that bind the vesicle and also the skeleton, cytoskeleton. ATP is added, phosphate is added to that motor protein that is going to move along that cytoskeleton tract, and every time you put in ATP molecule, it makes it a little unstable, and that vesicle and that motor protein move along the track. So ATP is not simply wasted once it's used up. ATP can actually be regenerated in the cell, and this happens in the mitochondria. So in our cells, we have ATP. That ATP is hydrolyzed in order to power our endergonic reactions. So ATP is used to power endergonic reactions. And what we're left with is an ATP plus a phosphate molecule that's inactive. We call it an inorganic phosphate. Energy is released, usually through that transfer of that phosphate molecule. But then we need to regenerate this ATP because we want to be able to use it again. So in an endergonic reaction, we rebuild our ATP by adding in a phosphate. So the exergonic breakdown of ATP is used to power endergonic reactions in our cell. That ADP can then be regenerated through an endergonic reaction to produce more ATP. Hopefully you noticed that I fixed a little typo there as I was talking. Now we have to put energy in to build ATP, but when we break ATP, that energy is released.